So welcome back, students. So let's wait for a few more minutes. So all will join, then we'll start. Okay, so now number is fine. So except one, all are there. Okay, so we can start. So first, we should discuss what your test was, and many of you scored very nice, and some were very poor. So it's better to discuss the answers, what is going to be the right answer, and what you have attempted. And you can see the first question was who discovered the restriction enzyme. So it's a simple question of history. So in this, the answer is the Nathan, Arbor, and Smith. So these are the right answers. So 22%, 20 out of 22 students answered very right, and 2 given a wrong answer for this. And the second question was an enzyme that take internal cut in the DNA R. So 21 answer very right. It's endoenzyme, yes, or also you can call it endonuclease. And one answer is wrong. So who is this one? I'm not naming him. So ligase, Joe of ligase is joining, not the cutting. Okay, so third question, it's some typological errors are there, not so. So question is up to this one only. The sequence recognized by restriction endonuclease is, so what is the property of that? A mirror sequence? No. Answer is a palindromic sequence. That is the right answer. Conservative sequence? No. Reverse complementary? No. All of the above? No. So answer right answer is the a palindromic sequence, because each restriction enzyme is going to recognize its own specific sequence. When you are saying the conserved sequence, means every endonuclease are going to recognize that. They are not mirror sequences. They are palindromics. Both the ends they are running same, reading same. 5' to 3'. That's the palindrome. And now move to the question number 4. Most commonly used restriction enzymes are, so most commonly are find type 2 are, so that's the right answer. 18 have given right answer. 3 are given wrong. These are type 1 and type 3. So right is the type 2, because this is the one which is having the specificity in the fluorescein cut. And then come to the single stranded unpaired extension formed by restriction cleavage is. So when a single stranded unpaired extension is there and it is forming after the restriction cleavage, so that is a sticky ends. So not a blunt end, no. Sticky end, yes. Flanking region, it's not the flanking region, flanking region is some. So none of the above and all of the above is not the answer. So right answer is the sticky ends. 19 out of 23, 22 have given right answer. Then we are moving to question number 6. So the enzyme from bacteria cleave viral DNA in defense against virus R. These are the restriction enzymes. That's why we have named it restriction because they are the part of defense system of bacteria and they are protecting the bacteria from virus infection. So the right answer is restriction enzyme. Endonuclease, everyone have the endonuclease, so that they are not called as a defense one. Ligases, they are not a defense, they are of the repair. And adapter sequence enzymes, so it's nothing, not such thing is there. So question number 7, which one are required by the restriction enzyme type 2? So the requirement of the type 2, we have divided the enzymes on the basis of the requirement also. And we in the table, if anyone of has seen the table, it is the requirement they need only the magnesium ion. So the right answer is magnesium and the magnesium ions. So this is the answer. So others are wrong. So 15 is given the right answer. So those who have given wrong, so answer is the magnesium and magnesium ion. And to add the nucleotide at 3 prime, the enzyme required is at 3 prime of DNA. You want to add a nucleotide, so enzyme required is the terminal transferase. It's not the DNA polymerase. So DNA polymerase it is for the replication. You need it can't extend at the 3 terminals. It can't add. It can add only when the replication is going to be there. But if you want to just add without a template, then the terminal transferase is the enzyme. Tag polymerase is the wrong answer. They both are the polymerases. Terminal transcriptase is also a wrong answer. So transcriptase is something which is going to form RNA over mRNA. So that's not the right answer. So right answer is terminal transferase. So 5 out of 22 have given the right answer. Rest all gave the wrong answer. So it is a terminal transferase. So in terminal transferase only which can add your nucleotide at the 3' end. And next question is the phosphatase and kinase are isomer, neoisomer, isopolymer, transferase, none of the above. They are different enzymes, so they have no relation. So only even the job they do the reverse the job. They are not the isomer. Neoisomer is the case of restriction enzyme. Isopolymer is also for the restriction enzyme. So the right answer is going to be none of the above. And so 14 has given the right answer. And 10th is the drawback of tag polymerase. It is not thermostable. Yes, it is thermostable. It requires ions. Yes, it requires ions, but it's not a drawback of it. Every enzyme requires ions. And it also have a nuclease activity. It's not a drawback. We do not have a nuclease activity. And the answer that is that it may incorporate wrong base. This is the drawback. We do not have the proofreading activity. That's why it is not. For all the application, you can't use it. This is the drawback of it. This is the limitation of this tag polymerase. It may incorporate wrong base. 14 have given the right answer. So 6 have given wrong answer. 2 haven't attempted it, I think. Okay, so this is the your answers. So whatever you, if any one of you want any more clearance out of your tests, please ask. Because after this we have to move to our next module. And today is just introductory one, so we are not going to go in detail of that. So we will just talk about what this module is going to be and what points we are going to be discussing this module. Okay, so any query that you would like to ask? Any question? Am I audible? Because
yes,
no one is responding, so that's why I'm asking.
no, sir, no doubt.
No, sir.
Okay, so poor performers, start focusing on your study, and even if you're sleeping during the class, at least if you are sleeping during the class, then how you can watch the recording? I don't think you will be interested in watching recording, but at least follow the syllabus. At least see the slides which are uploaded on the LMS. From where you can get, at least you are going to get the information So these internals are going to be a part of your final score also. So be attentive during the class, or at least be attentive during the exam, during your test. Give your test wholeheartedly, not just to appear in the test. Okay, it is only for your betterment, not it is not going to help me in any way. Okay, so now we have to move to our next part. So that is going to be the introduction to module number 2. And before moving to module number 2, I would like to take your attendance first, because then last somebody is who are not at all attentive, and those who are also not joining, they also came and claim the attendance. So just for a moment, I'm opening your attendance sheet. Okay, so Amazon is a bit slow, so I'm just calling your names. Just turn on your camera. Adish, Adish, Adish,
Present, sir.
Atoiba,
Present, sir.
Ayushi,
Present, sir.
Present, sir.
Demetrius,
Present, sir.
Ishita,
Present, sir.
Janvi,
Present, sir.
Kshitij,
Present, sir.
Manaswini, Namrita,
Present, sir.
Nandini,
Present, sir.
Nehal,
Present, sir.
Rupal,
Present, sir.
Sahiba, Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridev,
Present, sir.
Vivek,
Present, sir.
Yukta,
Present, sir.
and Yuvraj.
Present, sir.
So who is absent today? Which one who is absent? One student is absent. Okay, so Shit is left. Now attendance is done, so Shit is left. She's came back. Okay, so we have to share the syllabus. What the next is. Okay, so second, I'm sharing you the syllabus so that you can see also. Okay, so this is your syllabus, and second module is going to talk about mainly two things. One is the southern blotting. Simply, you can say what is the blotting, and second is the detection. And in the blotting, three types of blottings are there: southern blotting, northern blotting, western blotting. And detection wise, two types of detection. It is a radioactive and non-radioactive. And term that we are using, that we are going to do, we are using for detection, that is called probe or probing. So this second module is going to talk about the tools that we are going to utilize for detecting whether your recombination is going to happen or not. So whether your gene of interest is going to be there or not, or you have whether you what the main job that you want to perform, or the main aim of the recombinant DNA technology, the recombination is there or not. So for that, what techniques that we are going to utilize is going to be there. So again, the weightage of this module is going to be 10%. So 10% means out of your 28 three sessions we are going to dedicate for this. These two modules. Sorry, this module number 2. So we will finish. We'll try to finish this module in our 3 classes, as it is not a very large module. It's very small. Just mainly 3 topics are there. So just today I would like to tell you the thing that the term is there, the bloating, or also we commonly call it blot. And we can also use one more term that is called the transfer. So simply here the name that terminal that we are going to utilize, here we are transferring. Whom we are transferring? Here we are going to transfer DNA, and here we may transfer the RNA. We are going to, so two types of nucleic acid we are going to have. One is the DNA and second is the RNA. If you are transferring and where to transfer, we are transferring to solid spot. So if you are transferring your DNA to the solid spot, then you use the term southern blotting. And if you transfer RNA on a solid spot, then we are going to use term northern blotting. And one more term is called western. So if you are transferring protein, then we are going to use term western blotting. So these 3 types of blotting depend upon the what type of molecule you are going to study. Now coming to the name, how the name of these technologies are going to come. These techniques, why we call it southern, northern, western. The name of the southern the first discovered technique is your southern blotting, and here the name southern come from the name of the scientist who discovered this technique. But others, northern and western, they just come by. One is the southern and other is going to be northern, and third one is going to be the western. And if you're going to describe one more technique that is going to be related to the blotting and the transfer, and you are going to transfer other than DNA, RNA, protein, we may also use term eastern for that. So all the 4 direction we can utilize for it. And then coming to the probing. So probing means the finding, and you want what you want to find. So in case of DNA, we want to find our DNA sequence. In case of RNA is also a nucleotide, so RNA also has a sequence. So by RNA you can find the RNA sequence. Western blotting I mentioned, it is going to be protein. So you are going to detect the protein. So your detection is also going to be based on the protein only. So why two DNA may interact? Why two DNA may interact? Just a moment. Okay, so as I'm saying, why, whom to whom DNA is going to interact? Manaswini?
Yes, sir.
If you want to detect DNA, क्लास में बाद में करता हूँ. Okay, so if you want to detect, DNA is there. So what's the special property of the DNA? Can you tell me the properties of DNA?
Properties of DNA, sir. They have property of semi-conservative replication, and then they obviously we already know that they carry genetic material. There is a possibility for them to be modified, which is the basis of recombinant DNA technology, and then they are chemically inert. So there are many properties. I'm just telling randomly.
I would like to was waiting for only one property that you didn't mention. That property is the double stranded. And not just double stranded, the property one is the complementarity. So I was waiting for this word, complementarity, and so what it is. Can you explain this complementarity?
Uh, in let's say one particular DNA strand, you have four different bases. So when another DNA strand comes and winds with the starting DNA strand to form a double helical structure, then it is formed by hydrogen bonds um, two complementary bases. So the four bases are adenine, guanine, cytosine, and thymidine. So there's complementary base pairing between the nitrogen bases on two different DNA strands, and because of this, they are able to interact and form a double helical structure.
Yeah, so this is the main thing, the complementarity, as Manaswini was saying, that a complementary means the pairing of A with the C, A with the T and G with the C, and this pairing is universal, it is not going to be changed. Means if the complementary sequence is going to be there, then only two nucleotides or two nucleotide sequence are going to pair. Like if you are going to have a sequence AGTC, so if AGTC is there, then the complementary sequence to AGTC is going to be GACT. So if the another sequence is this one that I mentioned, if that is going to be there, then only this pairing is going to occur. Otherwise, no sequence will pair. Or a non-specific pairing may occur if some overhang that you mentioned, like if T is there, then T may pair with the A, but complete pairing is not going to be there. Okay, so this nature or this behavior of your DNA, that complementarity is there and there is universality in the complementarity. Similarly, this similar complementary behavior is there with your RNA also. RNA also form a double-stranded structure if it found a complementary sequence. Like for that, for many one of you, all of you said that DNA is single-stranded, DNA is single-stranded. Can anybody tell me whether the double-stranded form of RNA exists within you? I'm not asking about double-stranded mRNA, that RNA in virus. I'm asking a double-stranded nature within you, within a eukaryotic cell. So give a thought that where in a eukaryotic cell a double-stranded RNA exists. Okay, so I am giving you a time of I think 5 minutes will be more than enough for you to think where the double-stranded RNA exists in us. Okay, so think for 5 minutes, then I will explain it further. Okay, students, so I hope you have given the deep thought. So who will answer it? So where in us have the double stranded form of RNA? Manaswini, Yuvraj, would you like to say something?
In the cytoplasm.
Cytoplasm, where in the cytoplasm? Give the example where that RNA is double stranded. Name the RNA. Everything is there in cytoplasm only. That's a wise answer, you say that, or a diplomatic answer. Manaswini,
So my answer was also similar, just that it's used in some cellular mechanisms, right? So it must be found in the cytoplasm.
When I will answer, you will say, "Oh, you know it. You all know that." Nehal, Yukta,
Yes, sir.
so any idea?
No, sir. Actually, I also think the same.
Okay, so one more question I would like to ask here, in a similar question, similar note. So why RNA is not double stranded? Both are nucleotides. Adenine is there. Only sugar is different, and the double stranded formation is because of the base pairing. All the bases are alike. On base wise, your DNA and RNA are no way different. But RNA is single stranded and DNA is double stranded. Why we do not have double stranded RNA? So again, I'm giving you 5 more minutes to keep your thought on it. Then I will answer you both together, both the questions together. Okay. So give you again thought why we do not have a double stranded RNA. And if double stranded form of RNA is there in your cell, where this double stranded form of RNA is there? I'm saying you all know the answer of first. First question you all know. Okay, and just you have to revisit your RNA and types of RNA that you are having. So just if you revisit your RNA and different types of RNA, you will get the answer of the first question. And the second question also you will get the answer when you will visit how RNA is going to be formed. Okay, so I have given you the hint. So from where you have to get the answer. So now give a thought and come up with the answer. Your time start now. 5 more minutes is with you. I have given you the hints. Okay, so now who will answer, or no one would like to answer it? No one have answer.
Sir, wouldn't rRNA be found in the ER?
No, rRNA is not a right answer. It's a tRNA.
Okay.
So answer is the tRNA that you are having. So tRNA having the double-stranded form, and that double-stranded form occur in tRNA because of self-complementary regions are there. To the double-stranded one, we call them as a stem, and unpaired form in the tRNA is called a loop. So that's why that you might, if you remember the structure of tRNA, different stems and loops are there, different arms are there. So the stems and loops they form because of the self-complementary, and they form a partial double-stranded regions in them. And that's why one more property that you are amphi that we characterize, then you differentiate, and you want to know whether the sequence that you are having, it is going to be mRNA or it is going to be tRNA, that you can find on the basis of checking is there any self-complementary region is present on your DNA sequence or your RNA sequence or not. Okay, so this is a simple thing. So RNA can also form a double-stranded one. And second question was that why your RNA is not double-stranded. As I mentioned that if the complementary sequence or complementary strand is going to be there, then it may form a double-stranded one also. But the reason is that when the transcription is occurring, transcription occur only on one only on one strand of your DNA. The another strand of your DNA though do not transcribe. So only one strand of DNA transcribe. So in the absence of complementary strand, RNA remain single-stranded. But when the replication is going to occur, replication occur on the both the strands. So the complementary strand is always there and they remain paired. Wherever in case of the transcription, as the transcription is occurring, your DNA which was single-stranded, it become double-stranded. Okay, so no pairing remain between the DNA and RNA. Once this pairing occur, your mRNA is going to, your RNA is going to detach from your DNA sequence. Otherwise, during the transcription process, also your mRNA and DNA doublet is there. So double strand form is there when even during the transcription, because it is forming over the template of DNA. But the pairing of DNA is more suited rather than the DNA RNA. So that's why. And Manaswini and Yuvraj also answered one thing, that in some cellular process also your RNA may be double-stranded one. That is yes, that's also right. That you know the RNA interference or RNA-I mechanism. So in that mechanism, your RNA form the double-stranded one with the DNA sequence or with the RNA also. So if that double-stranded formation leads to the leads to your gene silencing. Okay, so this is the thing that exists. These are a normal thing, normal question, but for them you need to think. So if you because you all know the answer. I don't think any one of you are not aware about these things that I told you. A transcription occur on one strand that you all know. So second strand is not there transcribing. That's why the RNA can't form the double-stranded one. For the double-stranded structure, you need two strands. Simple. So nothing new I mentioned. And tRNA, you all know that in the tRNA, stems and loops are there. So there exists double-stranded nature within the RNA. But that double-stranded is not like the DNA that we are having. All the long stretch of DNA is double-stranded because of the complementary strand exists in DNA, which do not exist in case of RNA. So this is the just brief introduction that we because these two things we are going to utilize, the complementarity and transferring it to the solid sport. And one more thing I would like to mention when the term transferring is going to be there. So two things, they are interacting because of there are going to be some interaction. And that interaction in case of DNA, you know that is a hydrogen bond that formed between the nitrogenous bases. That is a hydrogen bond interaction. Second type of interaction that we call ionic interaction. So some charge is going to be there. Then with charge also, two things they can stay together. One is going to be negatively charged, other is going to be positively charged. So with that also they can held together. So these two, and one more property is there that's called the capillary action. Means capillary action means through the pores, small liquid and small things they can travel up. So the capillary action is another thing that we're going to utilize in this module. And electrophoresis also we are going to utilize in it in the southern blotting that we're talking. So these 3, 4, whatever the points I mentioned, you can read them again, get some idea about these points that I mentioned. That will help you in understanding the blotting. So how you are blotting and how blotting is going to help you further, and how this blotting is going to help you in the detection. The main aim of blotting is detection. That's it. So even in the diagnosis also you can utilize this blotting. And this interaction that I mentioned, this and the Western blotting that you mentioned, this Western one is a part of your immune things. When you are blotting the protein, so protein you cannot detect with the DNA, but the protein, for detection protein, you need a protein, because protein can interact, DNA can interact, DNA protein also can interact, but that is a different story. But in here Western blotting, we are going to utilize that protein interaction. And then the issue of the detection still remain, because the DNA is also blind. For DNA also we are blind, for RNA also blind, protein also we are blind. And even if you say the UV light you will use and ETBR you will use, then it cannot differentiate which DNA you want to detect. So we want to give our eyes. So our eyes, we want to producing our eyes in your this blotting. So for that term we used called probing. So we are going to use probe, and these probes are called DNA probe in case of DNA, RNA probe in case of RNA. And these probes are going to be labeled. So label you can see, and these label may be a radio label in case of radio labeling, and non-radio in case of non-radio labeling. It may be an enzyme in case of non-radio, it may be a fluorescence tag. So all these things we are going to use, and these things are going to help us in further recombinant DNA technology for when you want to go into for the analysis of your recombinant clone. Okay, so with this I would like to rest today's discussion, and formal module number 2 will start from Monday. Okay, since then, stay safe, stay happy, and anything if you like to ask, please let me know. And all are present except one. I have downloaded the list. I will check who is absent from the list. Okay, so till then, stay safe, stay happy. Good day.
Okay, sir.
Thank you, sir.
Sir. Thank you, sir.